Uh, Good afternoon, everybody. A very warm and hearty welcome to the next edition of the JMitra Scientific Webinar. Today's webinar is on a topic which is, well, it's a topic that affects all of us in today's scenario. And antibody testing in SARS CoV-2 infections and their implications in clinical practice is a topic that we feel is very relevant to the medical scenario today. And it is important that we all stay updated on the current changes, current practices, because treating of COVID-19, antibody testing with respect to COVID-19 is something that is an ever-evolving scenario today, right? Every single day, there are new discoveries made. And we are very pleased to have with us today Dr. Manoj Kahar. Dr. Manoj Kahar comes with more than 20 years of experience in the field of pathology and transfusion medicine. Having completed his MD in pathology from Government Medical College, Surat in 1999, he followed that up with a specialization in transfusion science from the University of Manchester in 2016, and has even completed his specialization in stem cell transplantation in 2020. He also completed a PhD from the Government Medical College in South Gujarat in Surat in 2014. Dr. Kahar is a very well-published academician, having been published at the both national and international level on various topics of pathology and transfusion medicine. We're very pleased to have you with us, sir, and we would like to take this forward. With your permission, we'd like to start with the presentation, and Dr. Kahar, over to you, sir.
Thank you, Dr. Arjun Mahajan, sir. I am very thankful to to Arjun Mahajan, sir, who is General Manager, JMitra. I'm also thankful to Mr. Pratik Kanani, who is Zonal Manager, JMitra Company Limited, to give me this opportunity to deliver this talk, this webinar. I am also thankful to all the delegates who have joined in for this webinar today. And the topic today for us is antibody testing in SARS CoV-2 infection and their implications in clinical practice. I hope I am audible to all of you. Am I audible, sir?
Yes, sir. Very loud and clear. And before sir begins his webinar presentation, my request is, in case anybody has any questions, please put it in the Q&A box or in the chat box, and we will endeavor to get all your queries and questions answered towards the end of this particular webinar. Over to you, sir.
Thank you. As the audience in today's webinar, they vary from students who are pursuing their MD or DNB, or those who have just completed their MD or DNB are in practice. So I've kept this lecture to a very basic, so it's apt to all attendees. Many may be senior to me and maybe well versed with the subject, but I'll stick to the basics. And I've taken into consideration that our audience may be some students who are pursuing their MD and DNB. So for few of the participants, it may seem to be a very basic lecture. So first, the topics what I will discuss today is structure of the coronavirus, the immune response to the SARS CoV-2 infection, the role of laboratory in SARS CoV-2 infection, timelines for various diagnostic tests for infection, the antibody kinetics, the types of the various serologic tests which are available for SARS CoV-2 infection, quality management and performance characteristics of serological tests, interpretation of these serological tests. Then we will discuss utility and limitations of SARS CoV-2 serological tests, impact of clinical specificity, clinical sensitivity, and disease prevalence on interpretation of serological test results, recommendations for reporting results of SARS CoV-2 serological tests. I'll provide you some templates how to issue the reports, and then need for orthogonal testing for SARS CoV-2 infection, and then we'll conclude. So let's begin. So as we all know, the severe acute respiratory syndrome, severe acute respiratory syndrome coronavirus 2, SARS CoV-2, it is a beta coronavirus discovered in China in December 2019. And we know it is responsible for an international outbreak of respiratory illness termed COVID-19, or we say coronavirus disease 19. And as all of us know, it is ranging from mild self-limiting respiratory tract illness to severe progressive pneumonia, multi-organ failure, and unfortunately death in some patients. It is now one of the top priority pathogens to be dealt with because it has high transmissibility, severe illness and associated mortality, wide geographical spread, lack of control measures with knowledge gaps in veterinary and human epidemiology, immunity, and pathogenesis. This is the structure of SARS CoV-2. And as the figure shows here, there are 4 structural proteins. One is the spike protein, this. And this spike protein is named as S protein. Then you have the envelope protein. Then we have the membrane protein. And then we have the nucleocapsid protein, or the N protein. These are the 4 main structural proteins: spike protein, envelope protein, membrane protein, and nucleoprotein, which is a nucleocapsid protein, as some authors describe, N protein. Now, the spike protein, it is divided into subunits S1 and S2. This S1 of spike protein, it is containing receptor binding domain, and this receptor binding domain, it is involved in binding with the human angiotensin converting 2 enzyme receptor and mediating host cell entry. S2, it facilitates fusion of the viral and host membranes, and distal region of the S protein, which is S1 RBD, it is the least conserved among members of the beta coronavirus. And this S1 protein, they are more likely to induce a SARS-CoV-2 specific antibody response. The N protein, on the contrary, is most abundantly expressed immunodominant protein among COVIDs, and its function is to stabilize the viral RNA. So, in the structure of the SARS-CoV, we have 4 structural proteins: spike protein, the membrane protein, the envelope protein, and the nucleoprotein or nucleocapsid. Now, each of these proteins, they have specific functions, they have specific roles. So, spike protein, it utilizes an N-terminal signal sequence to gain access to the cell. Then nucleocapsid protein, it binds the viral genome in a beads on a string type conformation. This envelope protein, it is a transmembrane protein with ion channel activity. Membrane protein, it binds to nucleocapside. And we have one functional protein, that is heme agglutinin estrase dimer protein, or HE protein, which binds sialic acids on surface glycoproteins. So these are the various proteins, what they do, and these are their role. So spike protein, it mediates attachment to host receptors. Nucleocapsid protein, it binds the viral genome to replicase transcriptase complex, packages the encapsulated genome into viral particles. Envelope protein, it facilitates assembly and release of the virus involved in ion channel activity. This membrane protein, it promotes membrane curvature. And this functional protein HE, it is thought to enhance S protein mediated cell entry and virus spread through the mucosa. Now, this figure explains us the various steps involved in the coronavirus life cycle. So, this coronavirus, it enters our pneumocytes through ACE receptor. Once it attaches and enters the pneumocyte, then there is viral RNA release, there is translation, RNA replication, all the structural proteins are being formed, then the proteins, they assemble, virions are made, and the virions are released. So this explains the viral cycle of the coronavirus. Now, when the virus enters the body, as we have discussed and as I mentioned earlier, the virus attaches to the ACE receptor via spike protein. And as I told you, there are two subfractions of S protein. One is S1 and S2. S1 is the protein which actually binds with the ACE receptor, and S2 subfragment, it helps in the fusion. And once the virus is in our body, our immune system, it responds. And as we know, the basic immune responses we have, there are two types of immune responses: innate immune response and adaptive immune response. So the adaptive immune response, it is either to the B cells or the T cells. And the B cells, they lead to the productions of various antibody molecules. So once the virus has entered our immune system, it responds by a basic innate immune response and also by adaptive immune response. And this immune response is to B cells and T cells. And as you see here, initially, the antibody which is formed is the IgM class of antibody. Then we have the IgA class of antibody which is following it. And lastly, we have the formation of IgG antibody. Our immune system tries to tackle this virus through various mechanisms. So one is we produce neutralizing antibodies. And these neutralizing antibodies, they prevent the attachment of coronavirus to our pneumocyte. They prevent the attachment of coronavirus to our pneumocytes. And these antibodies are neutralizing. The second way in which our immune system helps us is by the way of natural killer cells. So the natural killer cells, through antibody-dependent cell cytotoxicity, it kills the coronavirus which have infected the pneumocytes. So one mechanism to deal with the coronavirus is by producing neutralizing antibody. The other mechanism is through NK cell, natural killer cell, via antibody-dependent cellular cytotoxicity. The third way in which then our immune system deals with the coronavirus is through complement and complement-dependent cytotoxicity. This is the membrane attack complex. We know MSC C5 to C9, and they form a holes in the infected cell, try to kill the virus by complement-dependent cytotoxicity. And the fourth mechanism by which the coronavirus infected cells they are taken care of is by antibody-dependent cellular phagocytosis. So these are the four main mechanisms by which the coronavirus is being dealt by our immune system. Now, what is the role of laboratory in SARS-CoV-2 infection? So we have, as a laboratory person or as a laboratory, we have role at different phases of the SARS-CoV-2 cycle. So laboratory play a role in epidemiologic surveillance via RT-PCR or the anti-SARS-CoV-2 antibodies. Once the person gets infection, the laboratory plays an important role in diagnosis. And the diagnosis mainly is by RT-PCR. Then once the patient suffers from the overt disease, then also as laboratorians, we play an important role in prognosticating and therapeutic monitoring by offering various laboratory tests like CBCs or neutrophil lymphocyte ratio, ferritin, LDH, D dimers, interleukins. So here we also play a role. And once the person has recovered, then again we play a role by detecting the anti-SARS CoV-2 antibodies, and this can be useful for epidemiologic surveillance. So laboratory plays an important role in SARS CoV-2 during different phases of the virus. Once again, I am describing the role of laboratory. Mainly, we are involved in diagnosing the infection by nucleic acid amplification test for viral RNA, and the beneficiary of this test are the individual healthcare or long-term care facilities, public health agencies, and the other way we help are by detecting antibody detection, and the beneficiaries are those potentially immune to SARS-CoV-2, identify those which are potentially immune to SARS-CoV-2 infection. Healthcare facilities are the beneficiary. Experimental therapy is benefited by this antibody detection. Public health authorities are also benefited. Now, as I told you, the adaptive immunity is either by a T cell response or by B cell response. So in T cells response, we have CD4 response, CD8 T cells response, and in B cell, first there is expansion of the plasma blast, there is formation of the memory B cells, then formation of IgM antibodies, then IgA antibodies, and lastly, it is followed by IgG antibodies. So we have T cell response and B cell response as adaptive immunity to coronavirus. Okay. Now this figure explains us the use of diagnostic tests over a period of time. Say for example, before the onset of symptoms, it's difficult to diagnose this virus. Maybe in this incubation phase, if we check, we can detect either the antigens through nasopharyngeal swab or respiratory tract specimens, bronchoalveolar lavage sputum PCR, etc. Once the patient is having symptom, after the onset of the symptom, during the initial 8-10 days, the main way to diagnose is to diagnose the viral nucleic acid either through nasopharyngeal swab PCR or from isolating the virus from other respiratory tract samples, bronchoalveolar lavage sputum PCR, and some studies also say that stool can be used for PCR studies. So in initial phase, we diagnose the virus through detecting the viral nucleic acids. After, say, 10, 12, 14 days, the patient starts developing first the IgM antibodies, and then it is followed by the IgG antibodies, which is shown here in the green graph. So this is the timeline of how we used different tests during the SARS-CoV-2 infections. The same thing is being depicted in these two graphs, where in the initial phase, we are left with diagnosing the infection either by detecting the antigen or the viral RNA. And if the patient presents late in the disease course, then your antigen has already come down, your RT-PCR may be negative, and then you are left with detecting antibodies. If the person is giving us mild response, then your IgG may be not that high. But if the person or patient is giving a good immune response, then you will have a good IgG response. This green graph here shows IgM response. This is IgG mild response, and the other graph, golden yellow, what we are showing is a patient who is giving a good or strong response with IgG antibodies. Some patients, they are infected, but they do not produce antibodies to that extent which could be detected by the routine kits. So few patients, they are infected, but they do not have the antibody response which can be detected. The same thing is being explained here, where in the initial part of the disease, initial phase of the disease, we can detect the viral RNA or antigen, and later we can detect the IgM antibody and the IgG antibody. Now, when we see the antibody response, basically the antibodies, they are classified into two broad categories, and these are binding and neutralizing antibodies. These binding antibodies, they inactivate the virus through mechanisms such as complement activation or opsonization. And I've explained you earlier how our immune system helps to take care of the coronavirus. So binding antibodies, they act through complement activation or opsonization. Neutralizing antibodies, they inhibit by binding to regions of the virus that directly interact with the host cell receptors, effectively blocking viral entry and inhibiting replication. So what they do, these neutralizing antibodies, they bind to the part of the virus which are going to interact with our receptors. These are the neutralizing antibodies which bind to the regions of the host virus, bind to the virus which are going to interact with the host cell receptors. Now, we have many commercial serologic assays, and the serologic assays, they are available for detecting total antibodies. When we say total antibodies, then the kit detects all the three antibodies, IgM, IgA, and IgG. All the three antibodies are detected in this. These are kits for total antibodies. Then we have kits which detect specific subclasses, like for IgG alone, IgM alone, or IgA. We have kits also for detecting neutralizing antibodies using qualitative or semi-quantitative methods. However, at present, there is no clear evidence to support the clinical utility of standalone IgM test. And it has been said that IgA-based assays, they suffer from lower specificity as compared to IgG-based assays and are currently not recommended for use either by the Centers for Disease Control and Prevention or the Infectious Disease Society of America. So, detection of total antibodies is first. If you use such a kit, it may enhance sensitivity of antibody testing, and the other best option would be to go for a kit which is detecting IgG. Right? So there is no clear evidence to support the clinical utility of standalone IgM testing, and IgA-based assays, they suffer from lower specificity as compared to IgG-based assays and are currently not recommended. As mentioned earlier, in the initial phase of the infection, antibody detection is unlikely because during the initial phase of the infection, we can detect antigen and the RNA. And multiple published studies, they have demonstrated that most individuals develop an IgM, IgA, IgG response within 7 to 14 days of symptom onset. And with over 90% of the individuals, they are seropositive after 3 weeks. So if you see the timeline, after 14 days, more or less, we have the production of either IgM, IgA, or IgG antibodies. Now, as you see here, the IgM and IgA peak and decline earlier than IgG. So, your IgM, this light green one, and the IgA, they peak and they decline also earlier than IgG, and IgG antibody tends to persist for a longer time. But at present, we do not know for how long this IgG antibody will be present. IgG antibodies correlate with disease severity, decline at varying rates, may be detectable for months following infection. Please note that approximately 4 to 10% of the population with confirmed SARS-CoV-2 infection may have either an undetectable or delayed antibody response. This point is also to be kept in mind that about 4 to 10% of the population with confirmed SARS-CoV-2 infection, they may have antibody which is not detectable, or they have a delayed antibody response. Then there is this question of longevity of antibodies developed against SARS-CoV-2. So some studies, they indicate that up to 40% of confirmed individuals become IgG seronegative by the early convalescent phase, while others have demonstrated that antibodies decline yet remain detectable for months post-infection. We are not very sure how long these antibodies which have developed of IgG class will remain and provide long-term immunity. This is a question. Given these inconsistencies, the precise kinetics of the SARS-CoV-2 antibody response requires further elucidation. And antibody kinetics in specified subpopulation like immunosuppressed patients, cancer patients, other subgroups may differ, and they need to be continuously studied. So we are not very sure of the antibody kinetics in specified subpopulations like immunosuppressed patients, cancer patients, and other subgroups. Now, as mentioned earlier, majority of our serologic assays, they may detect either IgG or they may detect either singly IgG, singly IgM, IgA, or total. Total antibody means total of all the three. Most of the assays, they use serum, some accept plasma, and rest frequently whole blood. Currently, serologic testing is not recommended for other samples such as saliva, cerebrospinal fluid, etc. And in most of the serologic tests, the antigen which is targeted is the receptor binding domain of spike protein, followed by the whole of the S protein, full S, or maybe S1 subfraction, or maybe S2 subfraction. And few kits, they also target the antigen N. So the antigens used to detect the antibodies may be either the receptor binding domain, or maybe the full spike protein, or maybe the subfraction S1 or subfraction S2, or sometimes the nucleocapsid antigen N. And most of the current serologic assays, they are qualitative, with few being semi-quantitative. So most of the assays, they try to detect this receptor binding domain, which is present on the spike. Now, we have two types of assays. One is we are detecting antibodies by the direct binding ELISA principle, where we have the antigen coated, we add the serum, and then incubate. If there are antibodies, they will go to the antigen. Then we add the HRP conjugated anti-IgG, IgM. If the antibodies are not there, if they are not there, you don't have the low OD. And if the antibodies are present, you will get a high OD. This is our simple ELISA principle, direct binding ELISA. For those ELISAs which are surrogate for neutralization ELISA, neutralizing antibodies. These are surrogate for neutralizing antibodies. In that, we have a recombinant ACE2 plus biotin tag. To that, the antibodies are added. The unbound ACE2 is washed away. Then we add the HRP streptavidin plus the TMB substrate. If the antibody is not there, you will have high OD. If the neutralizing antibody is there, then you will have low OD, because the neutralizing antibody, it binds to the ACE receptors and do not allow this complex to attach. So this is basically the principle of surrogate neutralization ELISA. What is a neutralizing antibody? A neutralizing antibody is responsible for defending cells from pathogens, and they are produced naturally by the immune response. And neutralizing antibodies, they are produced by the B cells within the immune system, and they are characterized by stopping infections through incapacitation of the invading pathogen. So virus neutralizing antibodies directly inactivate a virus by blocking the cell entry or its replication or other viral functions. And neutralizing antibodies developed against SARS CoV-2, they prevent the interaction between the receptor binding domain viral spike protein RBD and cell surface receptor angiotensin converting 2 enzyme. So if you see here, this is our SARS CoV-2 virus. This is the spike protein with its RBD. And this is the neutralizing antibody. And this neutralizing antibody prevents the binding of this receptor binding domain to the human ACE2 receptor. So by this neutralizing antibody, the virus is being prevented from getting attached to the ACE2 receptor. The detection of neutralizing antibodies as such, it requires functional assay. And the gold standard for detecting neutralizing antibodies is the plaque reduction neutralization test, which is technically very challenging to perform. It requires viral and cellular culture. It is a prolonged turnaround time of about days to weeks, and it requires biosafety level 3 facilities, and is not a test to be implemented in routine laboratory practice. So this protocol for, I'm not going to detail of this. This is the gold standard test to detect neutralizing antibodies, and it requires stringent criteria. It requires biosafety level 3 facilities. So it's not practically possible in routine laboratory practice. Then the other option to this, to detect neutralizing antibodies, is this pseudovirus neutralization test. And it is also a cumbersome process. However, this test needs biosafety level 2. This plaque reduction neutralization test, it needs biosafety level 3, very demanding to follow. This is a little bit This can be done at biosafety level 2, but still it's a very cumbersome process. So to overcome the above challenges, alternative methods have been developed. And for that, we have blockade of binding immunoassays. And this blockade of binding immunoassays, they are adaptable to ELISA platform, right? So instead of these two testing protocols, this plaque reduction neutralization test and pseudovirus neutralization assay, we have an alternative, which is termed as blockade of binding antibodies. And JMitra offers you this solution. They have this kit, COVID-19 neutralizing antibody microELISA. So this is an alternative to or these are the steps where for performing this neutralizing. And as I've used the word, this is the surrogate neutralization ELISA. The recommended are these two tests: plaque reduction neutralization test and pseudovirus neutralization test. But as they are demanding, not practical feasible for routine laboratory practice, so there are alternatives, and one of the alternatives is blockade of binding immunoassays, and JMitra has this kit for this particular reason. These neutralizing antibodies, they are key to address the reduction of virus capture and spread of infection. In the presence of neutralizing antibodies, it will determine if the individual is having a good humoral immunity, if the vaccine is successful, and how many infected individuals have remained asymptomatic. And this is the test principle, right? Here, the receptor binding domain to ACE receptor is blocked by the neutralizing antibody. So because of the presence of the neutralizing antibody, this RBD is not allowed to bind to the ACE receptor. So in this case, COVID-19 neutralizing antibodies positive test. The neutralizing antibodies are present and they do not allow the receptor binding domain to attach to the ACE receptor. In this case, the receptor binding domain of the virus is successfully binding to the ACE receptor because this doesn't have the neutralizing antibody. This is the concept for neutralizing antibody ELISA-based assays. Then we have this point of care devices where there are lateral flow assays utilizing immunochromatographic chemistry to detect antibodies, usually point of care devices where we have the test pen, the control line. But the sensitivity and specificity of these tests are questionable. Then we have ELISA-linked immunoassays. We have fluorescent microparticle immunoassays. And this is the basic ELISA concept we all very well know. We have antigens coated on the well, then we add the serum, then we add the conjugate, then we add the substrate, and if the antibody is present, the conjugate will bind, substrate will bind, and then we have, after adding the stop solution, the reading is being taken, and you have the OD, and based on the OD, we say whether the antibody is present or not. This is the basic of a ELISA test. And JMitra, they have the test for various antibodies. This is the ELISA for total antibodies, where these IgM, IgG, IgA, all the antibodies, they will be detected in the same kit. And this is a kit only for IgG antibodies. So JMitra offers you 3 types of kits. One is this surrogate neutralizing antibody test, COVID-19 neutralizing antibody Microlisa, and then you have this total antibody IgG, IgM, IgA, and IgG Microlisa. Few companies, they have the kits which are based on chemiluminescence immunoassays or chemiluminescent microparticle immunoassays. And here, the principle remains the same, where you have the antigen coated, then sample containing the antibody, then HRP label, anti-human IgG murine monoclonal antibody, and then you have signal reagent with enhancer, which will luminesce, and the luminometer will detect the luminescence and tell you whether the antibody is present or not. When you search on internet, you will get many studies which compared the sensitivity and specificity of kits produced by various manufacturers. I am not going into detail of this, but the conclusion is that most of these kits for detection of antibodies, their sensitivity and specificity range from say about 92% sensitivity to almost 100%, and about 99% of the specificity to 100%. So most of the diagnostic tests available by different manufacturers, they have very high sensitivity and specificity. So now we go to another important issue, that is to evaluate the performance characteristics of serologic assays. Assessing the relative performance characteristic of each serologic assay is a little bit complicated, as the approach, if you see here, the approach is different. Some have chemiluminescence based, some have lateral flow assays, some have, so the approach is different. The sample size, sample collection time, disease prevalence in the population tested by each manufacturer will vary widely. And as laboratory professionals, we should take these variables into consideration when evaluating assay performance. So the quality management implementation, they say that QC must be identified, verified, implemented for routine SARS CoV testing based on test complexity and manufacturer system. A minimum of 2 levels of quality control, positive and negative, should be included with each run of the specified assay. For qualitative and semi-quantitative assays, a negative QC and a positive QC near the cutoff must be run at least daily. For SARS-CoV-2 serologic assays, controls may be provided as a part of the assay kit or maybe need to be sourced separately. And for the, if you want to source it separately, the laboratories can purchase separate controls provided by the assay manufacturer or third-party vendors, or can use pooled patient samples. Use of assay calibrator material as control is discouraged. But if needed, the calibrator material must be from a different kit lot. So you can't use the calibrator as a control in the first instance, but if you are compelled to do it, then you should use calibrator material from different kit lot to make your positive control. QC material should also match the analyte detected by the specific assay and patient matrix. Then there are lots of pre-analytical considerations to be taken care of, like sampling time, sample stability, as well as potential exogenous and endogenous interferences. The time of sample collection is important when selecting positive samples for verification studies. As we have discussed in the kinetics, most of the patients will show their antibodies after a period of, say, about 14 days of the onset of symptoms. So the timing of the sample collection is very important when you are selecting patients for making positive samples for verification. So in order to verify test performance at the optimal reported sensitivity for most current emergency use authorization assays, samples should be collected at least 14 days post-symptom onset or PCR positivity. And such samples only should be used for verification. And when you are using samples collected from patients less than 14 days, then it should be clearly stated. So the recommendation is for preparing positive controls, the samples should be collected at least 14 days post-symptom onset or PCR positive. For quality management implementation, 20 QC data points on separate days are used to determine the target control mean and SD to establish the range. If we are using vendor material assigned ranges, then the laboratory should verify the product. And the QC performance should be monitored in real time to identify SIPs and trends. Laboratories should participate in proficiency testing either using the vendor products or alternative assessment program. Now, as we know that COVID-19 is a recent disease, the kits have come into market in recent times, and all these kits, they are not extensively evaluated. And therefore, as laboratorians, we need to implement more rigorous quality management system until assay reliability is established. This is a new disease for all of us. The kits have been in the market for not very long. They are not extensively evaluated, and therefore, as laboratory personnel, we must implement more rigorous quality management systems until this assay's reliability is established. And this may include analysis of additional QC material, performing additional lot-to-lot comparisons, identifying a partner laboratory for more frequent sample exchanges. This all we can do. We have to look after the imprecision. The reproducibility and repeatability of the serological assay around the positive cutoff must be verified. And for qualitative assays, a positive and a negative sample can be used, and the positive sample should be just above the cutoff. Semi-quantitative assays should be evaluated as quantitative assays, and the samples could span low, mid, and high signal to cutoff with at least one sample near the cutoff. And the intraday and interday precision experiments should test both positive and negative samples over 10 replicates on the same day, or 10 runs on minimum of 5 days over multiple shifts. For lateral flow assays for point of care device, that QC must be assessed for interday only over 5 days with multiple testing operators. So you must give the test device to different technicians over 5 days. And these are the various guidelines for designing the imprecision experiments. Prepare positive negative controls, preferably at concentration where imprecision claims were made by the manufacturer. Run 5 times daily for 5 days. Calculate mean standard deviation coefficient of variation for repeatability and within laboratory imprecisions of the index values like cutoff index, signal to cutoff ratio, and compare against corresponding manufacturer's claims. And the imprecision should be fall within manufacturer's claim limits where available. Then we also need to verify the accuracy. And the accuracy is to be verified for single analyte or a total analyte. So single analyte means those kits which are detecting only IgG or only IgM. And total analyte means those kits which are detecting all the 3 antibodies, IgG, IgM, as well as IgA. A minimum of 10 negative and 10 positive samples per sample type should be used. And for total antibodies, it is optimal to use known positive samples from each antibody class. Because in total analyte kits, we are detecting all the 3 antibodies, IgG, IgM, and IgA. Accuracy verification must be demonstrated with known positive samples for each antibody class that could be reported for multiple differentiated analytes. And you need to have combination of minimum 20 samples. Like you should have sample which is IgM negative or IgG positive, both IgM and IgG positive, IgM positive, IgG positive, IgM negative, IgG negative. And should be assessed to class specific positive and negative agreement and to verify clinical specificity. And for SARS CoV, this particular sample is difficult to get, which is IgM positive and IgG negative due to concurrent seroconversion. So it is highly difficult to get such a sample which is IgM positive and IgG negative. Carryover. This is highly recommended to assess for carryover contamination by measuring a negative sample in duplicate, followed by a high sample in duplicate, followed by a negative sample in duplicate again. So you need to have 2 negative samples in duplicate, followed by 2 very high samples in duplicate, followed by 2 negative samples in duplicate. And the high samples should be within 10% of the upper measurement range. Carryover may not be required when the analytical platform uses disposable sample tips. And how we calculate? The calculate percentage of carryover is by average of duplicate negative sample after high sample divided by average of duplicate negative samples before high sample multiplied by 100. So this is how you calculate the carryover. And the carryover should be within the manufacturer's claim or less than 2 times the analytical CV of the cutoff of COI, cutoff index, or sample to cutoff ratio. So this is how you design the carryover. Then cross reactivity. It is not required, but recommended if resources allow. And if you are not verifying this cross reactivity, if it is not assessed, then positive results should be provided with a comment indicating possible cross reactivity as per manufacturer's information. So I'll not go much into the detail of this cross reactivity, but as it is not required, but if resources allow, you can do this cross reactivity design. And for that, you need to have samples which were collected before COVID-19 appeared. And it is important to note that cross reactivity is more likely to be demonstrated when the sample size per species subtype greater than 10. Interference is also not to be verified, not required, but recommended if resources allow, and in consultation with the available resources, prepare samples with common interfering substances like icterus, hemolysis, lipemia from patients with and without COVID-19. And the calculation is again as per the data as per the calculation which I have described in the cross-reactivity experiment. For semi-quantitative or quantitative serologic assays, the reportable range must be verified. And the verification should be done by using non-diluted known standards of anti-SARS CoV-2 antibodies. And such non-diluted known standards, now they are available from WHO. And if unavailable, if such a standard is not available, then we can use an alternate calibrator lot or patient samples that span the analytical measuring range. And future standardization of quantitative assays to single international standard will be essential for accurate measurement of antibody levels once a protective immunity threshold is established. Now we go to the interpretation of serologic tests. So majority of the serologic tests, they are qualitative in design, and they usually tell us whether the result is positive or negative. Negative results indicate SARS-CoV-2 antibodies were not present or are below defined detection limits. As I mentioned earlier, some patients, they produce antibodies, but not to that extent which are being detected by our routine tests. Negative results cannot rule out active or prior infection, and results should be interpreted in context of antibody classes detected, the antigenic targets, time of sample collection, disease severity, and assay analytical performance characteristics, which we have discussed earlier. Now, this is the interpretation of anti-S and antibody. That is, anti-S means against the spike protein, and anti-N means against the nucleocapsid protein. So you know that the patient is, you know the vaccination status of the patient. So if the person is vaccinated, and if both the antibodies are present, then the patient is vaccinated and previously infected. If the person is vaccinated, only anti-S antibody is present, anti-N antibody is absent, then the person is vaccinated and not previously infected. If the person is unvaccinated and both antibodies are present, that means not vaccinated and previously infected. And if both antibodies are absent, unvaccinated, not previously vaccinated or infected. So if you want to know this, we need to perform both the assays, anti-S antibody and anti-N antibody, and we need to know the vaccination status of the patient. If the vaccination status is unknown, then how to interpret? Anti-S antibody present, anti-N antibody present, previously infected, may or may not have been vaccinated. This antibody present, anti-N antibody absent, vaccinated with no previous infection. Both the antibodies absent, not previously vaccinated or infected. So with these two assays and the history of vaccination, we can come to some good conclusions. What are the utility and limitations of SARS-CoV-2 serology? One is it is supportive in diagnosis of COVID-19. See, in the first line I've written that SARS-CoV-2 serologic testing is not recommended as the primary approach for diagnosing of SARS-CoV-2 infection. We have to go for diagnosis by viral nucleic acid testing. This testing may be useful to diagnose COVID-19 in symptomatic patients presenting later in disease, that is after 9 to 14 days post-symptom onset, who test negative by a molecular test with optimal assay sensitivity occurring at least 2 to 3 weeks post-symptom onset. And as I mentioned earlier, total antibody or IgG testing may be more useful for evaluating patients presenting later in the disease course. Serologic testing alongside RT-PCR is also recommended to support the diagnosis of multi-system inflammatory syndrome in children because this present at a later date. And serologic testing should precede intravenous immunoglobulin or blood product administration, as these therapies may influence serologic tests. One of the utilities for supporting diagnosis of COVID-19. Second is convalescent plasma donor identification and manufacturing. This is at present out of vogue. However, with the serologic testing, you may identify samples which can be used for manufacturing positive controls, borderline positive controls. And the serologic testings are also useful for epidemiologic and seroprevalence studies. Arjun sir, can I take some 10-15 minutes more if you permit me?
Please go ahead, sir. Please go ahead. This is a very informative presentation. And, like you said, you're covering everyone from beginner to experts. So, there's always new things we can learn about COVID-19. And I would not like to break your flow right now. Please go ahead, sir.
So with your kind permission, I'll take just 10-15 minutes more and then end up. Okay. So clinical sensitivity, clinical specificity, they are influencing the interpretation of serologic tests. To minimize potential false positives and to be of clinical value, the CDC and IDSA have suggested using tests with clinical sensitivity and specificity of 99.5% or greater. And positive predictive values or negative predictive values, they will depend upon the disease prevalence in the target population and also on the assay clinical sensitivity and specificity. So they indicate % probability that a positive or a negative test result will correctly identify individuals with or without antibodies in a given population. So what I mean to say is, if the prevalence of the disease in the community is more, then the negative predictive value, the proportion of true negatives, it declines as disease prevalence increases. So if you see here, as the disease prevalence is increasing, the negative predictive value also true negative, proportion of the true negative is high. On the contrary, the positive predictive value is also influenced by the disease, is strongly influenced by the specificity of the assay when a disease prevalence is having a low prevalence. So when you are talking of a disease of low prevalence, then you have to select a kit of high specificity. So for example, there are two instances here. In the first instance here, the disease prevalence is high, 20%. So the disease prevalence is high, 20%, and a kit with a sensitivity of 95% and specificity of 95% will give you a positive predictive value of 82%, roughly 82%. If the disease prevalence in the community drops down to 5%, and the same kit with a sensitivity of 95% and specificity of 95%, see the positive predictive value. It has dropped down to 50%. So in this chart, if you see here, these are the true negative, these are the false positive, these are the true positive, and the false negative. If the prevalence of the disease in the community drop down, it is going to affect your positive predictive value. With the same kit, the sensitivity of 95% and specificity of 95%, the positive predictive value drops down from 82.6% to 50%. So how do you tackle this problem? For that, we go to orthogonal testing algorithms, which I'll discuss in the subsequent slides. So now we go to how to report the results. So results reporting. Clear and concise comments are needed to aid in interpretation of the serologic test results. And the qualitative nature of most serologic tests, it prohibits reporting of actual signal to cutoff values. So in our serologic reports, we should not report the actual signal to cutoff values because the assay specific values, they are not standardized. They do not indicate any actual concentration of SARS CoV-2 antibodies. Currently, no signal cutoff are available to correlate with protective immunity, and different dynamic ranges are available for current platforms. This is a general statement which we need to print in our all report, that serologic test is an aid in identifying individuals with an adaptive immune response to SARS CoV-2, indicating recent or prior infection. At this time, it is unknown for how long antibodies persist following infection. If the presence of antibodies confirm any protective immunity, SARS CoV-2 IgSS should not be used to diagnose acute SARS CoV-2 infection, and testing is limited to laboratories certified under this is for US. So US FDA requires that this 4 statements should be there in your reports. Now I'm giving you some templates how to report. I will not go into the details. I have given you the reference class at the end of the PPT, and there are very good guidelines which are available on net, which are free, which are given by CDC, then International Federation of Clinical Chemistry, and American Society of Microbiology. If you search the internet, there are very good guidelines which are given how to report the results of serologic tests for SARS-CoV-2 infection. So I'll not go in each one in detail, just for an example. For a negative, no IgG antibodies to SARS-CoV-2 detected. Negative results may occur in serum collected too soon following infection or in an immunosuppressed patient. Follow-up testing with molecular tests is recommended in symptomatic patients. This test should not be used to exclude active recent COVID-19 infection. This is an example template for reporting negative results. Similarly, for positive results, SARS-CoV-2 IgG antibodies detected, and results suggest recent or prior infection with SARS-CoV-2, correlation with epidemiologic risk factors and other clinical and laboratory findings is recommended. Serologic results should not be used to diagnose recent SARS-CoV-2 infection. Protective immunity cannot be inferred based on these results alone, and false positive results for IgG antibodies may occur due to cross-reactivity from pre-existing antibodies or other possible causes. So these are the templates. These are the guidelines how to frame our reports. Negative result. First line is negative. First line test is negative. The final result is reported as negative. No further testing required. Positive result. First line test is positive. Supplemental test is also positive. The final result is reported as positive. Discordant result, where your first line is positive and the supplemental test is negative. The final result is reported as discordant. So these are various examples. Same way. One last point, that is orthogonal testing. If a positive predictive value cannot be achieved, see here the positive predictive value is only 50%. In this setting, you have to go for orthogonal test. If a desired positive predictive value is not achieved, the CDC recommends use of an orthogonal testing OTA, a 2-step testing strategy where all initially positive results, they are tested with a second-line independent serologic test. Both the tests should have ideally have high sensitivities, more than 90%, or ideally the sensitivity should be more than 95%. And when you are selecting the test, the higher specificity test should be selected as first-line test to minimize the number of discordant results while still retaining optimal positive predictive value. And OTAs incorporating IgM or IgA serologic tests are not recommended, as there is higher likelihood of discordant results. When you are using this OTA protocol, you have to use either tests which detect total antibodies or IgG antibodies. See, this is one type of OTA. First test has sensitivity of 95% and 98%. The second test has a sensitivity of 99% and specificity of 95%. If you add both these tests, then the positive predictive value becomes 95%. If you see positive predictive value of test 1, it is only 49.2%. But when you add this both the tests, then the positive predictive value becomes 95%. From 49.2, it has jumped to 95%. Similarly, in this OTA2 example, we have test 1 with sensitivity 99%, 95% test specificity, test 2 sensitivity 95%, test 2 specificity 98%. The positive predictive value of test 1, this test, only one test, which was 28.8%, has jumped to 95%. So this is the use of orthogonal testing algorithm. So what are the recommended use for SARS-CoV-2? Now I am concluding the session. The serologic testings, they are support diagnosis in symptomatic patients who are presenting in late phase, or also in late complications such as MISC, multi-system inflammatory syndrome in children. Then serologic tests can help identify people who may have been infected or who have recovered from SARS-CoV-2 infection. Serologic testing can be used to screen potential convalescent plasma donors and in manufacture of convalescent plasma, but this practice nowadays is out of vogue. Serologic testing can be used for epidemiology and seroprevalence studies. Serologic testing can be used for vaccine response and efficacy studies. Limitations are false positive results may occur. Negative results do not preclude acute SARS-CoV-2 infection or viral setting. Serologic testings may not differentiate between natural infection and vaccine response. The durability and kinetics of the humoral immune response continue to be elucidated. And serologic results should not be used for determining individual protective immunity, return to work decisions, assessment of convalescent plasma recipients, use of personal protective equipment, assessment of higher genetic stock functions. This is not the role of serologic testing. So to conclude, though not recommended at first-line testing for diagnosis of SARS-CoV-2, serologic testing can play important function in the management of SARS-CoV-2 infection and pandemic. Clinical laboratories should use only serologic tests that have been developed and properly verified or validated in the clinical laboratory. And as laboratorians, we should recognize the utility and limitations of serologic tests and carefully select and implement emergency use authorized tests. And these are my references, and I would sincerely request you to read these top 3 references. They are very good guidelines on serologic testing, how to frame the reports, and how to counsel the patients. With this, I conclude and thank you a lot for your kind attention. Sir, over to you.
Thank you very much, Dr. Kahar. I must say that, COVID-19 has been around now for roughly about 2 years. And although we think we know a lot about it, every day there are some new developments coming out in this field. Every day it is important for academicians, for clinicians, for doctors to update their knowledge. So when we came up with this topic, many thought that what new will they talk about? Something new. But when I had that discussion with you last month, I had a fair idea that your attention to detail is so precise, so accurate that you've covered a broad spectrum in about 1 hour and 14 minutes, and you have going by the comments in the chat box, I can very seriously say that it has reached out and impacted the audience in a very positive manner. So thank you, sir. Thank you very much for your time and effort. because of the paucity of time, in case we will not be able to take question and answers today, but I would request the attendees that in case you have any questions, please email them to us at connect@jmitra.co.in, and I will, in fact, we will get them answered and revert back to you very shortly. Sir, due to the prevalence of the COVID pandemic, unfortunately, we cannot be giving you a certificate in person at this point. And Mr. Kanani will be in touch with you. The certificate will come, we will get it delivered by hand by Mr. Pratik Kanani. But for the present, I would request you to accept this little digital certificate as a token of our appreciation. Dr. Kahar, thank you very much, sir. And we look forward to continued association with you, and maybe future webinars in the future in the month of December when we do our Tide event. Thank you, sir. Thank you very much.
It's my pleasure, sir. I am so thankful to you, Mr. Arjun Mahajan, sir. Thankful to Pratik Kanani, sir. And thank to all delegates, participants who have spared their valuable time and attended this webinar. And many of them, they are asking for the PDF, and it's for them only. So I'll pass on the PDF to Mr.
We will get the PDF for all the people who attendees. We will not only send you your certificates, but I'll get the PDF of this presentation emailed to you as well. Thank you, everybody. Okay. Have a great weekend and all the best. Stay safe.
Thank you.
Thank you.